find here which are the genes that are transferred into the normal traditional rice. So two genes that are being transferred here. The first gene is phytoin synthase, which is isolated from the plant that is called as daffodil, that is why it is called as daffodil gene. The second gene is carotene desaturase. This gene is isolated from a bacteria, that is Erwinia bacteria. Whereas some others also say that, and some of the other authors, what they did or what they say is that even the third gene, that is lycopene cyclase, is also been the gene which codes for lycopene cyclase is also been introduced into the plant so that it is able to produce beta carotene. But later it is found that the normal traditional rice is able to produce the lycopene cyclase. Thereafter they stopped transferring the gene in order to produce the lycopene cyclase, and the source for this lycopene cyclase is again the daffodil. That means from the daffodil earlier two genes were isolated, that is one is phytoin synthase, another one is lycopene cyclase. Later after that, as the scientists came to know that in the normal traditional rice, as the lycopene cyclase is synthesized, later they stopped this, only they started isolating only the phytoin synthase, and that is one gene from the daffodil, and the second gene is from the bacterium, that is carotene desaturase. So the importance of this genes is that in the normal rice, that is white color traditional rice, these two genes are lacking. The genes are phytoin synthase and the carotene desaturase. So through the recombinant DNA technology, what is done, the gene is being transferred so that the plant is able to synthesize phytoin synthase gene, carotene desaturase. So what is the importance of this phytoin synthase means? Phytoin synthase is able to convert the compound, that is geranyl diphosphate into phytoin. So for this step the enzyme required is the phytoin synthase, which is totally absent in the normal rice. Whereas the second gene is the carotene desaturase. Carotene desaturase is important for the conversion of the phytoin into lycopene. So with the help of these two enzymes, what happens, geranyl diphosphate is converted into phytoin, and from phytoin it is converted into lycopene. Finally, by using lycopene, the enzyme that is lycopene cyclase is converted into beta carotene, that is vitamin A precursor, which is present in the grains or the rice grains which we consume. Later this beta carotene is metabolized by the human body in order to synthesize what, the vitamin A. So this is all about the pathway of the golden rice. And coming to the next aspects of the rice, how the gene is introduced into rice. So already we know two genes. So in this slide you can find, as I have already said, the vector used for the transformation is the agrobacterium. You can find the agrobacterium totally here. Agrobacterium has got its own DNA and it has got its plasmid DNA. It has got its plasmid DNA. So this is what the plasmid DNA and its main DNA. So first thing is we need to isolate this plasmid from the agrobacterium. So this is what, the agrobacterium. And also we can find here the plasmid is not entire, the plasmid is cut open. So the cut open with the REN, you all know very well, restriction endonucleases. With using restriction endonucleases, we need to cut open the plasmid. The purpose of cutting the plasmid is we need to introduce our desired genes. That means the genes which we were isolated from the bacterium and the daffodil. As you can, gene is isolated from the chromosome of another organism. Another organism means the bacterium and the daffodil. So here only in this you can find only one gene, but you say that how many genes are there? One gene and two genes. So two genes, say here is one gene and here is the second gene. Both the genes are ligated, and this is what it is, the plasmid. They ligated this in the plasmid, and then the plasmid is introduced into the agrobacterium through various methods. Now the agrobacterium contains its main genome and the plasmid along with its desired genes. And in the next step, what is done? In the next step, this agrobacterium, which is carrying the plasmid and the genes, are allowed to infect the plant. So you can find here, that is, I'm going to show you. So this is, say, the plant cell, and this is what the agrobacterium which carries your desired genes. So agrobacterium is mixed with the plant, say for example rice grains, which is nothing but you can consider it as a plant cell. Agrobacterium is agrobacterium is infecting the plant cell. Agrobacterium is infecting the plant cell, I tell you. So you can find this is the agrobacterium cell and it is infecting the plant cell. So later what happens? After the infection, the agrobacterium, when it infects the plant cell, what happens? It starts multiplying inside the plant cell. While multiplication, it also multiplicates its plasmid, or it inserts its DNA into the plant DNA, that is plasmid DNA and plant genome, so that multiplication of the plasmid DNA along with the plant DNA occurs. After the multiplication, or whenever that is during replication, the plasmid gets integrated into the genome of the plant. Thereby the chance is that our genes may get incorporated into the plant genome, and later the plant cell multiplies. Thereby you can find the multiplication of the gene also. But the rate of transformation is very slow, in that, say for example, if you go for transferring about 1000 plants, hardly you may end up with about, say, 3 to 5 plants having our desired gene. So this is all about what this is the procedure for introduction of our gene into the rice. So once again I am going to repeat you. So this is what the agrobacterium. From the agrobacterium, what is done? The plasmid is isolated. Plasmid is cut open by using enzymes, and in this plasmid the desired genes, say gene 1 and gene 2, are ligated here, and next the plasmid is introduced into the agrobacterium. Agrobacterium is allowed to infect the plant cell, and here the bacterium is infecting the plant cell. After the infection, what it does, it introduces the plasmid along with your desired gene into the plant cell or the plant genome, and thus the plants are said to be transformants. So in this case we call them as what? Transformants. Transformants means the cells which have received our desired genes. So this is all about what the procedure for the introduction of the gene. And the last in